Welcome everyone to join our bioinformatics and genomics seminar today. And we're glad to invite Dr. Yoseph, Bhoj, and Dr. Joseph Barash from University of Pennsylvania, talking about their recent papers regarding the RNA splicing variants. Very exciting about that. Welcome. And Dr. Wen-Han Tang from BCH will co-host this meeting with me. So as usual, we will introduce our digital science education page. And for this year, we have 2 more seminars. And the next seminar will be November 17th. And the topic is about automated prediction of CNVs. And recently, we already updated this page. So what the previous seminars were are already uploaded to YouTube. So if you miss any previous talk, and if you are interested, you are welcome to come here to watch the YouTube here. Okay, with that, I will hand it over to Dr. Wen-Han Tan. Thank you, Wen-Han.
All right, thank you. So I have the great pleasure today of introducing our guest speakers for this series. Today we are delighted to have Dr. Elizabeth Bhoj and Dr. Yoseph Barash, who will be doing a joint presentation. Dr. Bhoj, who has been my friend and collaborator for many years, graduated from the University of Texas Southwestern Medical Center with MD and PhD degrees, and subsequently trained in medical genetics at Children's Hospital of Philadelphia, and followed by a molecular genetics fellowship. She is now an assistant professor in the Department of Pediatrics at University of Pennsylvania, and has published extensively on various syndrome delineations. She is very well funded through the NIH as well as various foundations, including the Chan Zuckerberg Initiative and other grants. And we also have the pleasure of having Dr. Yoseph Barash, who is an associate professor of genetics and of computer information science at the University of Pennsylvania. Dr. Barash graduated from the Hebrew University of Jerusalem with degrees in computer science, physics, and machine learning and computational biology, and has given many talks over the last several years on RNA informatics. He is a real leader in this field, and we are delighted to hear from him. And some of you may be interested to know that in Dr. Barash's younger days, he was a major in the Israel Defense Force and led an operational wing unit in an Air Force unit. So with that, we will hand over to Dr. Bhoj and Dr. Barash.
Hi, thanks, Wen-Han. I really appreciate it. So this invitation means so much to us, and we're so happy to be able to present together. We were co-PIs on this project, and so we thought, since no one's paying for us to fly anywhere, we can have both of us come and be part of it. So I'm going to be presenting the first part, and then Yosef will take over, and then we'll jump back to me for a little bit. And I hope that this is the usual way things go, but I wanted to say that both Yosef and I are completely happy to take questions during the talk. So please interrupt. And for the moderators, if I can't see that there's a question in the chat or anything, just definitely interrupt me. Okay, so Yosef and I are going to be telling you about our project tools and evaluation for RNA-seq splicing quantification for suspected Mendelian disorders across tissues. And he's at Penn, and I'm primarily at Children's Hospital of Philadelphia. And our big question when Yosef and I started talking was, how do we improve molecular diagnosis of Mendelian disorders? Like Wen-Han mentioned, I'm a clinical and molecular geneticist, and so this is basically the question that I ask every single day when I'm at work, when I'm at home, when I'm in the laboratory. As you all know, the basic standard of care that we have now, exome sequencing, we only get an answer in about 30% from all comers. And as we've added on genome sequencing, I think there's been some disappointment in the field because that hasn't really given us the diagnostic bump that we had hoped that it would. And one of the major challenges, no matter what modality you're using, if you're looking at DNA-based technologies, is how do you interpret non-coding variants, which of course are generally thrown out during most of the analyses pipelines. So one way to get around that problem is to, instead of looking at DNA-based diagnostics like exome and genome sequencing, to look towards non-DNA-based diagnostics as the next step in this process. So we can look on one step down and look at a more functional platform and use RNA-seq to look for RNA splicing aberrations directly rather than having to guess them from a DNA-based technology. So when we're talking about RNA splicing aberrations, what do we mean? So here we have our traditional set of exons and introns. And of course, as you all know, due to alternative splicing, we can end up with multiple different RNAs and multiple different proteins that result from the same DNA sequence. And so you can see here we have an example of alternate splicing leading to one product, another physiologic RNA splicing event leading to a different product. But you can also have a pathologic splicing event due to say a non-coding DNA change here. In the third example, then that leads to a pathologic change. So here you can see from the same DNA, we end up with two products, and then when we have an inclusion leading to a aberrant transcript. And of course, this is then going to have functional consequences. And of course, on your campus, you have many of the leaders in this field and many people that have already published extensively on this. And it's really been shown over the past 3 years that RNA sequencing is a useful tool for improving Mendelian diagnosis. And of course, papers that have come out from the Broad and other groups. And so what we posited is, how can we build on this idea that already exists, that we can use RNA-seq to enhance the interpretation of the non-coding variants, specifically the non-coding variants that are going to affect splicing changes. So let's say that we look in a control subject and we have a one splicing event that's going to give us a traditional splice donor, splice acceptor. However, we have now in this intron a de novo acceptor that's going to create a novel splicing, a novel splice site here. And so you say, oh, well, this is, when you put it in these slides and you make the boxes, you make it seem so easy. Why aren't we doing this already? However, there's additional challenges that come along with the DNA-based diagnostics. And Yoseph is going to tell you about those.
Thanks, Elizabeth. So I'll switch now to yeah, see it?
Perfect.
Good. Okay, so you know, why are we not doing this? So you know, how do we analyze RNA sequencing for aberrant splicing? You know, the challenges are that we need a method that would capture very accurately complex and de novo splicing variations. So Elizabeth just showed you one cartoon for de novo splicing. By de novo, I mean it's unannotated. We don't know about this until we find it. And we're going to talk about what complex splicing variations are in a second. We need some statistical formulation for how sure we are that there is a real change, or what the magnitude of the change is, and how sure we are that this is indeed something that is like a serious outlier that we can quantify safely and accurately. We need some way to visualize this, because this becomes complex very quickly, and I'll show you a few examples. And we need to correct for confounders. So for example, Elizabeth said, you know, in her cartoon, which of course makes it look easy, she said, let's say I have a control and that's what I see. But of course, what we actually have is a whole bunch of controls, those normals that maybe we're comparing against, and then we're comparing our patient against. But of course, those controls can have a lot of variability in them. And some of them is biological, but maybe some of them is the result of confounder effects that we need to correct because they were done in different labs and different times, maybe on different sequencing machines. And if we don't correct for them, we're going to introduce variability that will hamper our ability to actually get that quality calls for things that are significantly changing. And I'll illustrate that on one data set. Prioritization, of course, when we find all this stuff, we need to somehow prioritize this. We're not going to talk about this too much today. And there is a huge computational challenge because if I need to process hundreds and hundreds of controls and have this maybe as a data set to compare against, and then I need to process more and more patients as they come along, I need to do this efficiently. I don't want to redo everything every time. Okay. So let's step back a bit and think why this is challenging and how we might be able to do this. Because, you know, you probably read a lot of papers saying, you know, we quantify expression and we quantify maybe splicing or isoforms expression from RNA sequencing. But a lot of those methods that maybe you apply in your lab for other things will actually not work very well for the kind of diagnostics that we want to talk about here. So why is this? So let's look at this cartoon here. We have a diagram of a gene with 6 exons from left to right. And as you hopefully all know very well, RNA sequencing reads, what they will do is they will produce little fragments, little short sequencing of RNA from a high throughput sequencing Illumina machine. And with a mapper like STAR, for example, we will map them back to the genome or the transcriptome, depending on how you run it. And so these are those little tiles that you see here. Can you see my pointer or?
Yes.
Yes. Okay. So those are those little tiles. And the reason they are colored here is because in this cartoon, we assume they all came from different isoforms that are represented by colors. But of course, they don't really come with colors. So when those little reads map to, say, this first exon, they can come from different isoforms, but we actually don't know where they came from, which makes it a hard inference problem. Now, a lot of the algorithms that you use to quantify expression and to quantify isoform expression, Callisto, Salmon, you know, the differential expression with VSeq2, all of those things, in general, what they do is they assume they have a transcriptome, they assume they know the transcripts, and then they map those reads to the different exons and then quantify what they think might be the level of expression. The problem with that, of course, is in the first sentence, as I just said. They assume they know the isoforms. And what you're looking for here are things that are likely non-isoforms. There are de novo, there are cryptic splice sites that have been introduced. They are not associated with known ones. And we don't know where they start, we don't know where they end, and we still need to detect them. So those algorithms are actually not going to work for them. If they're doing pseudo alignments and they're assuming a transcriptome, they will never quantify that isoform because they've never seen it before and they don't know how to quantify it. So that's a problem. But luckily, we have other options. So another way to quantify splicing is by looking not at the entire isoforms and trying to assume we know what they are, but looking at what we call local splicing variations or splicing events and quantifying them. And for that, using what we call junction spanning reads. So here are examples for junction spanning reads from the red isoforms. And, you know, some of them are highlighted. We're focusing in here on the interesting part, which is exon number 2, marked in yellow, connected in the red isoforms to exon number 3. These are the highlighted red junction spanning reads. That's why they're like chopped in the middle. And maybe we also have reads from the blue isoforms that go between 2 and 4, and the green one that is maybe de novo, maybe it introduces cryptic splicing that was never occurred, not supposed to happen because maybe we jump from 2 to 5. That, you know, leads to leaving out important protein domains, and then the gene is not going to function properly. Okay, if you're on our story. So what we're seeing now are junction spanning reads. that go either from 2 to 3, 2 to 4, or 2 to 5. And a lot of other algorithms don't assume they know the isoforms, so they can't really give you isoform, which is unfortunate. But what they can give you is more accurate quantification for the relative ratios and the detection of those de novo isoforms. Again, some of them would assume known transcriptomes, so they're not good for this, and many algorithms do this even for splicing events. But some algorithms, like the one we developed in our lab, which I'm going to describe next, can enable you to detect all the de novo junctions, de novo exons, and all this complex splicing variations. So in this specific cartoon, what we're going to get out of our algorithm, which is called Magique, is a cartoon that is representing splicing between 2 to 3, 4, and 5. Each connection gets a color, and based on the junction spanning reads, there's some Bayesian algorithm, which I'm not going to go into, that will give you some posterior probabilities for the inclusion, the percent spliced in, or psi, in our language, which is going to be something between 0 and 1, for each one of those junctions, the red, the blue, and the green. And maybe sample 1 is our patient, and maybe sample 2 represents controls or a whole bunch of controls, and it's very different. So the algorithm will show us both of them and will show us what's called differential splicing, the changes. And maybe that's the one that we're after. Okay, so that's in a nutshell how we approach it, and that's why some algorithms are just, extremely useful for other things, but not for this clinical diagnostics. So the way we do this to quantify the de novo and complex splicing events is through this algorithm called Magique that we published in 2016. And the basic idea is very simple. Just let me explain it to you in one slide. If this is a gene splice graph, well, those rectangles are the exons and those arcs are the splicing events that we can detect through junction spanning reads, just like I showed you in the previous slide. Every time we find a split in the graph out of an exon, like you see here on the left, we call it a single source LSV, and we can quantify those ratios between those junctions. Or into an exon, like you see here on the right, that's called a single target, okay? And why is this useful for us? Because the LSV language, this local splicing variation language, can enable us to quantify all of those events that you see here on the top right, which have been studied in the splicing community for decades, since RNA splicing was found by Phil Sharp and other groups in 97.7, got a Nobel for this, of course. And so these have been studied for decades, but what we found is over a third of the transcriptome variations in humans are more complex than those simple binary ones, and they can involve multiple junctions and multiple exons, like the examples you see here on the bottom. That's why we call them complex, because they are not binary. They don't have just two options. They might have multiple junctions and multiple exons involved. And Magique can capture those, including de novo junctions, including de novo introns, retention events, and de novo exons. So to summarize, the tool we published in 2016 is able to find those complex splicing variations, de novo junctions, exons, introns. It has this built-in visualization that I showed you or illustrated a bit. It has built-in primer design. So if you want to validate this, you can basically copy paste this into another tool that we published in 2017. It will give you automatic prime-to-bizarre for your RT-PCR validations. And we've shown that it has very high accuracy and reproducibility compared to other tools. So that was back in 2016. So what's the problem? The problem is that we needed to develop Magique V2, which is some of those features we already released, and some of them are coming up, hopefully in a preprint in the next couple of months. But the difference is efficiency and scalability, right? So the original tool was just was targeting biological replicates. You're, designing an experiment, you have 3 replicates here, 4 replicates there, etc. What we need through this kind of analysis is to handle hundreds and hundreds of controls that we want to compare against, and maybe many different tissues, for example, GTEx for other projects in our lab as well. So you need something which is very efficient and scalable, and also the ability to do something we call an incremental build, right? So if you have a database of normals and now you have another batch, you need to normalize that batch, but you don't want to redo all those hundreds of samples. That's going to cost you a lot of time and a lot of money. You just want to do this incrementally, and that's an important feature that we added in Magique version 2. The other thing is to support, in terms of the statistical data analysis, heterogeneous data sets, because these are no longer biological replicates. These are humans. They, have different reasons to die. They have different ages, gender, etc. You have to accommodate for this in your statistical model. And related to that, you need to be able to correct for confounding factors. And for that, we just released a new algorithm called Moccasin that works with this. And I'll show you one slide showing the benefit of that. So just to show you what it's able to do, and this is the result of many, many people in the lab that has worked on this in the last 4 years that are shown here on the left. So this is GTEx, and to show you the efficiency, GTEx version 7, 1200 almost RNA sequencing samples on a single desktop with 12 thread, took 12 hours to bleed the splice graph that I just told you, and 18 hours to quantify all the splicing variations in them. And since then, we of course processed all of GTEx version 8, which is about 20,000 samples very efficiently. So that's the sort of power or the jump in capabilities between that enabled us to do this. And everything that Elizabeth is going to show you later is based on this, because now we can do this, right? Before that, we were just limited. Here are illustrations of the accuracy of this compared to other tools that are usually used to quantify splicing variations. We're showing here RMATs and Leafcutter that is also being used for clinical applications, for clinical diagnostics, and SUPA and WIPIT. These are tools that can handle large data sets, and that's why we're only comparing to them. So this is 3 cerebellum versus 3 muscle from GTEx. This is real data, and what we're measuring here is reproducibility. So basically we say, I don't care how your algorithm works, give me the set of events that are changing, and let's take another 3 cerebellum tissues, another 3 muscles from GTEx, and see if these are reproduced. So of course you want to be here at the top right, all the way at 100% or a ratio of 1. Every time, the first 100, 200, 300, they're always reproduced in that same order. Of course, that doesn't happen. We can see here that our tool has significantly higher reproducibility compared to all the other tools. And you can see here the difference between the previous Magic and the new one, because the new one enables more statistical other kinds of tests that we're not going to get into. So it enables us to maintain the same reproducibility, but detect more. And this is just 3 versus 3. We can do this for 15 versus 15. Again, this is the 2 versions of Magic versus the others. This is just one illustration of reproducibility. In the RNA field where I come from, people always want to see how well you're quantifying splicing compared to the gold standard in the field, which is RT-PCR done in triplicates. And we've done this for multiple conditions, 4 different conditions. This is one of them, has about 50 of those events. So think of them, each one of them is done 3 times. And this is Magic here, and the correlation between Magic and the gold standard, the RT-PCR versus Leafcutter. We've done it for all the other algorithms, I'm just not showing it, but the same results hold. We compare very favorably for that. Okay, so I haven't gone through, we've also done synthetic data sets and etc. The bottom line is we compare very favorably in terms of the accuracy of the algorithm, but there's other issues, okay? And one of the main issues when you want to handle large data sets and then do comparisons is that you need to correct for batch and confounding factors. And this is a data set that is not specifically related to this talk about clinical diagnosis, but it is very much about disease and problems of confounders that you would find in public data sets that you might be using in your lab. Okay, so this is the target data set for cancer in pediatrics, and has been used by I don't know how many labs all across the world, has hundreds of samples. This is their ALL samples. And what we found, which we haven't seen reported anywhere, is a huge difference between the samples in patients that have been sequenced by one machine, HiSeq 2000, and another machine, HiSeq 2500. Now, why is that? We don't know, right? But it is definitely there. So what you're seeing here is we take psi quantifications, the splicing quantifications for all of those, this is several hundred of those samples, and we embed them in a 2-dimensional space using UMAP. and we measure the amount of variation that is associated with the sequencing machine. And you can see that number here, it's almost 60%, okay? And furthermore, what we're annotating here is the same patient done multiple times on different machines, and you can see that they land in different places. And it doesn't matter if it was primary or relapse, what really determines the location is the sequencing machine. That's very sad, and that's troubling because people are using this from all over the world. Right? So we developed an algorithm called Moccasin, and I'm not going to go into the details, but it, combines very well with Magic. It takes its output, corrects it, and puts corrected output, so then you can sort of seamlessly continue with your analysis for the clinical pipeline or whatever else you want to do with it. And here is the corrected version of this data set after running Moccasin. Okay, you can see now the mixing very nicely of the two sequencing machines. You can see for the same patient that relapse and primary cluster very nicely and separately. And if you're wondering why we have a cluster on the left and a cluster on the right, that's because we have two cell types there that we separate. And that's a signal that we actually want to maintain. And you can see the association of signal associated with sequencing machines drops to 0.3. Okay. So this is just showing you examples of why this is not as easy, or hasn't been done before, in terms of getting things to be accurate, have a proper statistical formulation, having good visualization, correcting for confounders. Again, we didn't talk about prioritization, but obviously it's an important topic for clinical diagnostics, and then computational efficiency. But even if we can do this, what should we be sequencing? And that's where we're going to switch back to Elizabeth to talk about that.
Thank you. Okay, stealing the screen back. Okay, and so when Yoseph and I started this project, we realized moving RNA-seq into any kind of clinically relevant setting, one of the major questions that hadn't been asked before was how can you decide if any of the tissues that you can collect are going to be useful to do for RNA-seq, and if so, which tissues should you perform RNA-seq on? And the reason that this is such an issue is, so you can imagine when we do DNA-based technology, assuming that you're not having mosaicism, pretty much every single tissue that you can get should give you more or less the same DNA, as long as we're not talking about cancer or mosaicism. However, of course, that's not true for RNA-seq data. And there had been people that had looked previously to see, can we use commonly used tissues in terms of expression? And there was multiple pieces of literature that had already addressed with the expression issue, but there hadn't been a structured way of looking how splicing would have that be differential in different tissues. And so you can imagine if, say, we're using fibroblasts for a relatively readily available clinical sample, you would be able to see a normal splicing event in the fibroblasts, and then if it's also expressed and you see that splicing, then you'd be able to pick it up. So that leads us to the question of how do we measure this tissue specificity, specifically of splicing in the RNA-seq that we would be using for it. And so, like I said previously, gene expression had been dealt with, but splicing hadn't been addressed in a robust way before. And if you think about this clinically, and not just clinically for patient-centered, but also clinically based research studies, you're only going to be able to perform RNA-seq on clinically accessible tissues, which we termed cats. And so we're not talking about these cats. These are all Barash and Bhoj lab cats. But instead, we're talking about the tissues that you can easily get from a living donor. And so those would be whole blood, lymphoblasts that are then created as a cell line from the whole blood, or fibroblasts taken from a skin biopsy and then grown. So the major question of this paper was, can we quantify for the first time the limitations of these clinically accessible tissues, looking at pathogenic changes that primarily affect other tissues, if you're looking at neurologic disease or cardiovascular disease. And as I mentioned, expression is one of the major and already looked at issues. And the one that we really wanted to focus on was adding benefit back and saying, how much does tissue-specific splicing also affect those? And so drilling down to it a little more specifically, our major question for the paper was, when is splicing in non-cats, so in the brain, in the heart, and other tissues, inadequately represented by splicing in the clinically accessible tissues? How often can you use these tissues that you can access as a proxy for these other tissues? So you can imagine a couple different scenarios. Say we're interested in a neurologic disorder, and this is the typical splicing that we see in the controls. In our patient's brain, however, we have this non-coding variant that leads to a novel splice acceptor, and so we end up with a novel splicing product. If we look in the fibroblasts now from the same patient, in the controls, we're going to see the same pattern. But what we would hope is that we would be able to use the fibroblasts to again pick up this aberrant splicing pattern. However, you can imagine that in fibroblasts and controls, this particular gene is not expressed in fibroblasts. And so whether we look in the controls or the patient, we're not going to be able to see any expression of this. And so it's not going to be able to tell us anything about the aberrant splicing that we see because of the patient's variant. You can also imagine a situation where the fibroblasts have a different splicing pattern. And this is a situation that hadn't really been quantified before, where you do see expression of the gene, but the splicing pattern that you see in your clinically accessible tissue is actually different than what you see in your tissue of interest. And so, as you can imagine here, if you were to do RNA-seq on this patient's fibroblast, because this splice acceptor isn't used in the fibroblast and it is used in the brain over here, you're going to miss this variant that causes the novel splice acceptor. So you can see here, this would inadequately represent this tissue, and we wouldn't be able to find this variant on an RNA-seq sample of fibroblasts. So in order to do a formal quantification of how often this happens, we looked for splice junctions that were consistently used in non-cats, so that were used in brain, heart, and that were either unused or not quantifiable, usually because of expression, in clinically accessible tissues. And so we used the wonderful resource that already exists, looking at GTEx samples, and process 762 RNA-seq samples from GTEx. Just a little bit of background, there's 53 tissues that have been collected from recently deceased donors. Of those, 50 are non-cats. Those are the brains and the hearts. And then the 3 cat tissues that we had already decided to use were luckily already represented within GTEx. And so remember, those 3 cats are blood, lymphoblastic cell lines, and fibroblasts. That gives us about 15 donors for each tissue, and identified the junctions, again, that were used in the non-cats, greater than 50% of the time, and then that were unquantifiable or lowly used in the cats, so less than 10% of the time. And using those cutoffs, there were about 50,000 consistently used junctions in every non-cat tissue. And I'm not expecting you to be able to see everything on this. What I really want you to use this graph for is more of a gestalt. So on the y-axis here, we have a list of all of the tissues that are present in GTEx. And then on the x-axis, we have the number of splice junctions that are present. So this is more to give you a sense of how many per tissue. If we overlay on that, I
see a question, Elizabeth. What are the cats? Somebody's asking, can you go slowly over what are the 3 cats?
What are the 3 cats? Sure, the 3 cats are fibroblasts that we can get from skin biopsies from the patients. whole blood, which is the tissue that is most often used for clinical testing, of course, and then lymphoblastic cell lines derived from whole blood. And we know that often whole blood and lymphoblastic cell lines have similar profiles, but we had some interesting findings from that. Does that answer the question?
I think so.
Okay. Sorry, I can't see the chat, so I'm happy to be interrupted.
Laurie says yes.
Perfect. So if we overlay on top of that, the cats, so these are the brains and the hearts and the other tissues that we don't have access to, the tissues that we're going to be using, we now overlay the orange on top of them in the tissues that we do have access to. It turns out that about 17% of those junctions are poorly represented by at least one cat. So in about 1 out of 5 times, you need to be thoughtful in making a decision about what that's going to be. And up to about 10% of those junctions are actually poorly represented by all 3 cats. And you can see that there are some outliers here. There's a little bump in these tissues that are not represented by any of the 3 cats. And this bump, this is alphabetical by the tissues. And so this little bump here is actually all of the brain samples. So it starts with brain amygdala, and then the biggest bump that we have here is cerebellar. We also have an outlier down here, and this is down in the T's, and this is testes. So that was reassuring to us because it's been known that the testes has a lot of splice junctions that aren't present in other tissues, as well as a lot of brain-specific splice junctions. So that followed what we expected from this. And then when we look and drill down to it a little more closely, it turns out that there was about 13,000 genes in every non-cat tissue that aren't represented by any of the cats. And so when we looked a little bit further about how we should use that for clinical service, for clinical findings, what it told us is that what we should do is actually break it down and say, well, what should we be thinking about? Should we be thinking about this on the expression side, or should we be thinking about it on the splicing side? Maybe a lot of this is just being driven because these tissues have low expression of these genes, and that's why we're not seeing it. And so what you can see here is breaking down by all the tissues and then saying if we look across all 3 of the cats and how often are for each of the tissues that are we see the expression for, and we don't see it in the cats. And so you can see that there is a variable but significant proportion of the genes for each of the non-cat tissues that we see low expression for. But when we look a little bit more closely, it turns out that about only 4% of the genes are going to be expressed at a TPM higher than 10, and about 41% are going to be expressed higher than a TPM of 1. So what does that mean for our splicing, which is really important? That means that a lot of this effect that we're seeing is actually not due just to the low expression from them. It's actually due to splicing differences that we see within them as well. And so you could say, well, these are, if there's that much difference, if they're not similar splice forms across different tissues, maybe this isn't very clinically relevant. Maybe these are genes that we just don't really care about as clinicians and as looking for Mendelian disorders. And so as an overlay on top of this, we went through and pulled out the genes that are poorly represented by the cat tissues. And it turns out that 30% of them have already been annotated as disease causing, either in ClinVor or HDMD. And so we really goes to show that this does have clinical implications. And this is something that we need to be thinking about as it goes into the clinical sphere. And so, of course, the big give at the end, people say, give us the punchline. If we're just in general going to be picking one tissue to do RNA-seq on, what should we pick? And it turns out that perhaps not unexpectedly, fibroblasts generally perform better than whole blood or lymphoblastic cell lines for looking across all the tissues. And the averages are here. You're going to miss across all tissues about 10% in fibroblasts. 17% in lymphoblasts, and 34% if you use whole blood. So the tissue, if you're going to be looking across all represented tissues, would be fibroblasts to use. But the problem is often you don't care about looking across all of the tissues, right? A lot of people are cardiovascular researchers, or they're neurologic researchers, or they're trying to study expression differences in the gut based on a tissue sample they're taking. And so I think at this point, I go back over to Yoseph.
Sure.
And he's going to tell you about the MagicCat tool.
Right. Let me just switch.
Oh, sorry, I didn't stop sharing. That's my fault here.
Yeah. You can see it?
Perfect.
Okay, great. So yeah, basically we built this tool. Our full stack developer in my lab, Bhoj, will help Joseph, the student who led this study, build this together. And the idea is to give, the clinicians a tool to ask for specific, let's say disease-associated genes for a specific disease or for a specific tissue of interest. Are they going to be well represented? Which ones are going to be well represented? And that's basically what you can submit as a query into this MagicCat tool. You can select which tissues you care about for representation as the non-cat tissues. You can select predefined sets of genes that you can see here on the left. You can also upload your own set of interesting genes that you want to query, and you're going to get answers about which ones are well represented and not, and how well with splice graphs and etc. So hopefully you can put it for good use for whatever application you're doing, studying specific disease. So it's not just numbers, just like Elizabeth showed you before, but actually informs your research and helps you select which cat to use and know better whether is that using RNA sequencing in a cat, is that a viable option for you for treating patients or for finding stuff. Okay, so just to summarize what we've talked about so far, RNA sequencing is one approach for improving the molecular diagnosis, and we wanted to quantify challenges of RNA-seq in clinically accessible tissues for representing changes across other tissues. And so we use Magic, we used GTEx for that, and we found splicing in around 1,300 genes in each of the non-cats is not adequately represented by cat, and up to 40% of genes are not represented well despite being expressed in those cats, and about 30% of these genes are annotated as disease-causing. So that is something we want to take into account when we use RNA sequencing on cats to try and diagnose things. Now, what we haven't talked about is, what we started for. We want to do clinical diagnostics. And, in the first part, I told you how Magic is a wonderful tool that we've added a lot of features. So you'd say, can you give me a pipeline now? Right? You've shown me which tissues I should be using and when I can use them. That would be the MagicCat part, right? But can you also use Magic now to just give me a pipeline so I can diagnose, I can prioritize, etc.? And the answer is we're working on it. That's the short answer, and I'll show you some samples of this. Because of course now we have to adopt Magic not just to look at changes between tissues in GTEx, like I've shown you as a proof of concept, and to quantify accuracy, etc. We just need to turn it into a complete pipeline for diagnostics. And this is something which is, this is unpublished, this is results, and this is still ongoing in the lab. So this project we call MagicClean, if you like, or MagicClean. And the idea is to compare splicing in patients and many control samples. So we have a whole bunch of controls, and we have our patient, and we look at changes in splicing. So in this case, if you look at the percent splice inclusion, those diagrams on the right, you can see that really all the controls are really using the green one almost 100%, while our patient completely flipped, and they're using the inclusion of the blue exon completely. So they're completely flipped between control and this patient. That would possibly be something very interesting. And of course, then we will have to prioritize it based on, how big is the change, which gene is that the gene that is interesting for us? Is this affecting, a specific protein domain? Is that causing a frame shift or a pre-termination codon, etc.? Okay. And so in this specific case that I'm illustrating here, maybe in the patient, it's actually the novel cryptic splice site that is activated, something we've never seen. But again, Majik identified in the patient. That would make it even more interesting and probably would shoot it up in terms of prioritization of something we should be aware of and thinking about, because this is, maybe causing a complete disruption of the protein domain, or maybe causing degradation at the protein level. And so, GTEx, we have about 1,000 or even more samples to compare, and we have one patient. So this is sort of one versus the rest. That's kind of the statistical setting that we have here. And, as a first test, just for sanity and also to see how well Majik is doing on this, we took previously published cases. And this is an illustration of one case from the Cummings et al. Paper, which is one of those papers that, Elizabeth flashed in front of your eyes when we started. And this is an illustration from their paper. And you can see sashimi plots with numbers and illustration of the splice graph and the location of the mutation. This is straight from figure 2A in their paper. What we want is a pipeline that produced all of this automatically for you, and that's what we're working towards. In this case, and all the other cases, by the way, in the Cummings et al. Paper, we were able to automatically identify them and prioritize them as one of the top, I think, 5 scoring ones per patient or something like that. So very doable. So this is control, and this is patient D7 going through our pipeline, identifying this de novo splicing. You can see that green arc with tons of reads supporting it. And this is the changes in splicing. And again, we did this for all the cases in Cummings just to see that we can do this. But what about larger scale, right? Because these are a handful of patients. How can we evaluate that we're actually capturing interesting signals on a large scale? How do we even test this? Because how many cases do you have like this? How many papers and how many cases that have been studied and validated? So one interesting idea to do this from a preprint for Meredith et al. Is to measure enrichments for rare splicing, rare splice region variants in genes with outlier splicing. And so the idea is very simple. We look at something big like GTEx, and we find that rare sample that have a huge change. And then we say, overall, we should see an enrichment if we look at, say, their whole genome sequencing or their whole exome sequence in rare variants in that region that might be explaining it. Of course, we don't know, but we should see an enrichment. So the more enrichment of those that we see of those rare variants, the more possible explanations we're finding for those. So, hopefully a better method will be able to detect more sure, strong splicing changes in outliers that would be enriched in those rare variants. And so that's basically the kind of metric that they suggested, and we follow that as an idea and compare to their results. These are preliminary results, so take it with a grain of salt that we did. And the x-axis here is the splicing difference that we're seeing between the rest and that specific sample across 49 tissues. So every point here is a tissue, and a total of 14,000 samples, right? And so this is what we're seeing. If there's no difference, the difference is 0. That's the one on the left, all the way to 20% changes. And of course, as you can imagine, the bigger the difference is, the higher enrichment it is for rare variants. And this compared to what they showed in their method, compares very favorably. But of course, we need to make sure that we did it exactly like them. This is still a preprint in them, and this is still work in progress with us. So this seems to be very promising as preliminary results for the tool, but we're still working on this.
So sorry to interrupt, Joseph.
Yeah.
I think, okay, you're the last
yes,
slide, right?
That's
okay,
it.
great. All right.
So to summarize, identification of aberrant splicing is a way to improve diagnosis, and there are still challenges, including selection of tissue and efficient analysis. We developed new tools for clinical diagnosis, MagicCat that helps you explore which of the accessible tissues should you be using, and MagicClean, which is still in progress to actually do this as a pipeline. And thank you all. This is the link to the tool. This is, really the result of this collaboration between me and Elizabeth's both labs coming together that enabled this. We're also interested in collaborations, questions, and feedbacks, or tools, or data sets, or things to analyze. So please reach out. And these are all the people that did all this work, led by Joseph Eicher, a talented MD PhD GCB student who is joint between my lab, was literally jointly mentored between my lab and me and Elizabeth. Also got an F30 from an ICHD to do this. And we also got other grants for this. And this is the full stack developer that helped with other people from the lab. And thank you for your time.
Thank you. Thank you, Yosef, and thank you, Elizabeth, for the interesting talk. And we have two questions. One is from Mary. Mary, can you unmute yourself and ask a question?
Yes. Are all the references in your slide to journal articles from the same journal? And if so, which journal is it? Thank you.
I'm not sure I understood.
Do you mean the MagicCat paper? Where's the MagicCat paper been published? That it's if so, that's Genetics and Medicine. I think July or August.
All right. We have second question from Christy Murphy. Christy.
Yeah, hi. I was just wondering if you had looked at any fetal samples like placenta or amnio, and if you had, were those direct samples or had they been cultured?
Yeah, so that's a great question. So actually, when Joseph presented this work at the Genomics of Rare Disease conference last year, when he was in the middle of making it, that was one of the big questions that came up is, well, it's, what we really care about in a lot of Mendelian disorders is developmental disorders. And so he did include in addition to GTEx, he also included a fetal brain and a fetal heart expression library. And so those, if you use that is both in the final published version of the paper and then also the tool. So you can use, you can look at your genes of interest or your list of interests in the fetal brain or the fetal heart.
And if you have other data sets, Christy, that you would like us to include in the tool, shoot us an email and say, hey, this is a very interesting data set. If you could include this so I can see how this compares to CAT, and I don't want to run your Magic, and I don't want to do this analysis, just if you can just add those, we'd be happy to do that, right? So we have all the, all the machinery, point us to the data set, and we'll just add it to the tool, because eventually the tool is meant for you guys to use.
Yeah, I mean, that's such a fact. We've never, I mean, I've never considered amniocytes as a clinically accessible tissue, but of course, that's done far more often than skin biopsies for fibroblast are. So I think that would be a fascinating addition.
Great, thank you so much. I appreciate that.
Okay, I actually have one question. I think I'm going to maybe try the Magic. So maybe this question for the Yosef.
Yeah.
So it's kind of opposite because recently we have one patient and one control sample RNA-seq, very high coverage data, and we are also interested to find whether we, have any, novel splicing site. Because based on the RNA-seq analysis, we identified, the differential express gene. We do the pathway analysis. We find like one of the most significant pathways, like transcript misregulation. So that's maybe really related to the splicing. So I was just wondering, like for the MAGIC-2, I know MAGIC-2 is mainly for the scale data. So is that okay for one patient, one sample is working?
You can always run it. It's just way more efficient than the previous version. There's no limitation that MAGIC version 2 would not work for this. And the old algorithms are still there and still apply. You'll just get better performance. It will take way less memory, and it will take, it will be, if it's just 1-in-1 is right.
Yeah. Great.
Yeah, and it will tell you all the de novo splicing variations that it can detect, and it will tell you, it will quantify, big changes and have all the splice graph associated with them. You can click, and it will, if you have your tracks on the genome browser, you will see, your raw reads in them, so it will go back to that. Yeah.
Awesome, thank you. I don't think that we have any more question. Oh, we have one more question, sorry.
Yeah, I saw the fetal brain question. So that, if I remember correctly, it actually fell kind of within the range that we see from the different adult brain tissue. There's, I can't remember, 11 maybe different separate adult brain tissues in GTEx, and it actually fell within the range of the other adult, which was interesting. But you can, if you have any specific aspects that you want to look at it, you can play around with it. It's the app is very easy to use. You don't need any special skills. It's easier than using Windows. So you can, pull up fetal brain as your tissue of interest, and you can pull in HGMD pathogenic. What do these look like in fetal brain? It's really interesting to fool around with it.
Thank you. That's very exciting. Thank you.
I see another question here.
Yeah, sorry.
Okay. Yes. Yeah. Please
I
ask.
can ask it. I'm a rheumatoid.
Because CH164166 is not very.
That's my internal ID, unfortunately. If you can hear me, my question was really, can the MAGIC software identify, and I may have just missed this, like novel splicing events between an annotated exon within a locus and an exon that's not annotated, or say in an adjacent gene where it wouldn't necessarily presume that these are typical splicing events? Can that be inferred in these in those reads you evaluated that's exon spanning reads?
Yes. So the answer is sort of yes with an asterisk. And why? So MAGIC can detect de novo junctions and de novo exons, and it can detect connections between those, right? So basically your question is annotated exon to a cryptic exon, and the answer is yes, which is one of the big advantages of this, including also the de novo introns between them. So retentions, which is, I think, is pretty unique in that landscape of tools to do this like efficiently and robustly. But why am I saying with an asterisk? Because you said something about neighboring gene, right? And so if you're looking at trans-splicing, if you're talking about splicing, caused by mutations where you have like gene fusion, and that is not something MAGIC is currently built to do. So it will look for, it will basically, the way the software works is you give it a gene annotation, unlike some of the other tools. You will give it a gene annotation, and then it will use it as a base and supplement all the de novo stuff into it, right? And the reason we do this is basically, if you're not willing to take a gene annotation, you're going to miss out on all the known biology that we know about those genes. So we see this as a huge loss, because then it's harder to quantify, and this is a longer discussion. But philosophically, we think this is important. Now, this is how the tool works. But if you're talking about, for example, gene fusions, if you're talking about splicing between completely different genes, and that breaks that structure, and that is currently not something that we're doing.
Thank you.
Welcome.
All right, I don't think we have any more questions. With that, thank you, Inespes. Thank you, Yosef. It's a great talk.
Thank you all so much.
Thank you, everyone. Thank you for having us.
Thank you. Bye.
Bye.